picture this. You're hiking deep in the mountains, the air is crisp, the pines are swaying, and you come across this crystal clear, just perfectly pristine mountain stream.
Oh, yeah, the classic picturesque scene.
Right. You've been hiking for hours, you're thirsty, and the water looks like it literally belongs in a bottled water commercial. So naturally you take a sip. I mean, what could possibly go wrong?
Well, a lot, actually. It serves as the ultimate cautionary tale about trusting your senses.
Exactly. And that's why today we're doing a deep dive into exactly what goes wrong when that idyllic mountain water is harboring a microscopic threat. We're looking at two infamous single-celled gut parasites, Giardia and Entamoeba.
Yeah, and that pristine water, which is completely devoid of any visible cloudiness or sediment, it might just be hiding some of the most, incredibly resilient evolutionary hitchhikers on the planet.
Okay, let's unpack this.
Hmm.
Because in the clinical research you brought in today, doctors often group these two pathogens together simply because they present similarly in a clinical setting.
Right. A patient shows up with severe gastrointestinal distress, and the immediate diagnostic question is just, what protozoan is making this person sick?
Exactly. But from a biological and evolutionary standpoint, putting Giardia and Entamoeba in the same category is, well, it's like confusing a shark with a dolphin just because they both happen to swim.
That is a great way to put it. They belong to entirely different evolutionary lineages,
Mm-hmm.
and they attack the human body using completely distinct mechanisms.
Right.
Yet they have both somehow managed to exploit the exact same vulnerabilities in our environment and, for a very long time, in our medical science, too.
And this deep dive is going to trace their impact from 17th century microscopes all the way to modern DNA sequencing, which I promise will fundamentally change the way you look at a simple glass of tap water.
It absolutely will. But to understand how these pathogens sneak into our water supplies, we first need to establish what they actually are.
Right. And how they wage war once they reach the human gut. Because the clinical presentation depends entirely on which evolutionary path we're actually dealing with.
Exactly. So let's start with Giardia, which is, it's notorious for having an identity crisis right out of the gate.
Oh, yeah. The literature is a mess. You'll see Giardia lamblia, Giardia intestinalis, and Giardia duodenalis used almost interchangeably.
Right. And just to be clear, all three of those names refer to the exact same organism. It's just a byproduct of a really messy historical taxonomy.
So what is it biologically speaking?
Biologically, Giardia is a flagellate, so it uses these characteristic whip-like flagella to swim through your gastrointestinal tract.
Just swimming along until it hits its start.
Exactly. It swims until it reaches the upper parts of the small intestine, specifically the duodenum and the jejunum. But the crucial detail about Giardia, and this is really important, is its relationship to the host's tissue.
It doesn't invade, right?
Right. It does not invade. It doesn't burrow into the intestinal walls at all.
Which, superficially, that sounds like good news to me. You hear non-invasive and you assume, oh, it must be a milder infection.
Yeah, you would think so, but the mechanism of disease is incredibly insidious.
Yeah.
Because it relies on mechanical and immunological sabotage.
Sabotage how?
Well, the lining of the small intestine is intricately designed to maximize surface area for nutrient absorption, right? You have all these tiny microvilli.
Right, to soak up the nutrient.
Exactly. But when Giardia arrives, it uses this specialized ventral disc to basically suction cup itself to the microvilli. And as the parasites multiply, they essentially pave over that delicate lining.
Wow. So they just cover it completely.
Yeah, they physically coat the absorptive surface of the gut. And at the same time, they trigger an intense inflammatory response that blunts the microvilli and completely disrupts the host's digestive enzymes.
It's like throwing a massive heady tarp over a field of solar panels.
Oh, that's a perfect analogy.
Right. Because the panels are perfectly intact. They aren't physically broken, but they are completely blocked from absorbing sunlight.
Exactly. And because that tarp is blocking the absorption of crucial nutrients and fats, the body's entire digestive chemistry just gets thrown off.
Because the gut literally can't do its one job.
Right. The gut cannot perform its primary function, which leads to a severely malabsorptive clinical picture.
And that's what causes the symptoms, right?
Yes. This manifests as profound bloating, weight loss, and these highly characteristic greasy, foul-smelling stools.
Because of the fat.
Exactly. The stools float because of all that unabsorbed fat content.
Wow. Okay, so it stays on the surface, ruining the local environment. But honestly, that surface-level blockade is practically benign compared to the sheer violence of our other subject today.
Oh, definitely. Entamoeba operates on a completely different level of tissue destruction.
So how does Entamoeba move? It's not a flagellate, right?
No, Entamoeba is a sarcodine, so it moves and feeds by altering its cellular shape to push out these temporary extensions called pseudopods.
Like a classic amoeba blob.
Exactly. Now, there are at least 6 species of this genus that regularly colonize the human gut as harmless commensals. They just hang out.
But there's one that doesn't just hang out.
Right. Only one is a true tissue-invasive pathogen, and that's Entamoeba histolytica.
And the name itself is a dead giveaway for its mechanism, right?
Oh,
I mean, histo means tissue, and lytica means dissolving.
you nailed it. Tissue dissolving. Unlike Giardia's surface-level tarp, E. histolytica actively breaches the mucosal lining of the colon.
It just burrows right in.
Yeah, it secretes specialized enzymes that literally break down host tissues. It burrows into the intestinal wall to create these distinct flask-shaped ulcers.
Ouch. And that causes the bloody dysentery you read about.
Absolutely. And in severe cases, the amoeba penetrates deep enough to actually enter the portal bloodstream.
Wait, it gets into the blood?
It does, and from there, it can travel to other organs, taking up residence and forming massive necrotic abscesses in the liver, the lungs, or even the brain.
Okay, I want to pause on that specific complication for a second.
Yeah.
Because the phrase amoeba in the brain immediately triggers public panic.
Oh, for sure. Summer news cycles love that phrase.
Right. So we need to clearly separate Entamoeba histolytica from the famous brain-eating amoeba that generates those headlines.
Yes, very important distinction. The headline-grabbing organism is Naegleria fowleri, which is an entirely separate, free-living genus.
And how do you catch that one?
Infection with Naegleria fowleri happens when contaminated warm fresh water is forced up the nasal passages, like from jumping into a lake.
So it goes up your nose.
Right. That allows the amoeba to migrate directly along the olfactory nerve to the brain. It has zero fecal-oral transmission.
Got it. Whereas Entamoeba histolytica is strictly an intestinal parasite, right? Contracted through contaminated food or water.
Exactly. So if E. histolytica ends up in the liver or brain, it is strictly as a secondary systemic complication of a really advanced gastrointestinal infection.
Okay, that clarifies it. So we have Giardia, the flagellate that throws a malabsorptive tarp over the small intestine. And we have Entamoeba, the tissue-dissolving amoeba that actively destroys the colon.
Right. 2 vastly different modes of attack.
But they're both fragile, single-celled organisms that absolutely require a warm, wet human host to survive.
They do, which makes you wonder how they spread.
Exactly. The sheer mechanics of transmission present a massive biological hurdle. How do they survive the cold, harsh, nutrient-deprived environment of a mountain stream long enough to infect the next host?
Well, despite their vast evolutionary divergence, they actually arrived at the exact same morphological solution to environmental stress.
Which is?
The cyst.
So when they're actively feeding and multiplying inside the host, they exist as fragile trophozoites.
Right. But as they travel down the digestive tract and sense the environment changing, they start experiencing dehydration and a lack of cholesterol, they undergo this radical transformation.
What do they do?
They initiate a process called encystation. The parasites secrete this incredibly dense, complex wall. It often incorporates rigid biopolymers like chitin, along with specific glycoproteins, to completely encapsulate the organism.
So they effectively build a microscopic hibernation bunker.
Exactly, a bunker. Passed out of the body in feces, these dormant cysts suspend their metabolism and can survive for weeks, sometimes even months, in freezing water.
Just waiting to be swallowed by a hiker.
Just waiting to be ingested by a new host. And once they are, the stomach acid actually triggers them to hatch back into active trophozoites.
That is wild. And the structural integrity of these cyst walls, that led to one of the most significant and shocking public health crises of the 20th century.
Right? You're talking about Aspen.
Yes. The Aspen, Colorado outbreak in the winter of 1965 perfectly illustrates this danger. You have a booming ski resort town with about 1,200 residents and visitors.
Right.
And suddenly, over 10% of them, 123 people, come down with severe explosive giardiasis.
It was a massive localized epidemic.
Yeah. And the epidemiological investigation traced the outbreak to town water that had been cross-contaminated by leaking sewage lines. But the detail that just stunned public health officials was that the water was heavily chlorinated.
Yep. And standard municipal chlorination, the exact chemical levels we rely on to eradicate bacteria like E. coli and viruses, is fundamentally useless against Giardia and Entamoeba cysts.
Wait, so when I drink chlorinated city water, it's not the chlorine protecting me from Giardia?
Not at all. That chitin-reinforced sisqual is so dense and chemically inert that routine chlorine concentrations just bounce right off it.
Wow. If scientists and engineers discovered in 1965 that chlorine didn't penetrate these cysts, that raises a massive question about municipal infrastructure.
It did. It threw a huge wrench in the gears.
Were water treatment facilities just relying on a false sense of security because, I don't know, overhauling the system to physically filter the water was too expensive?
Well, cost and infrastructure inertia definitely played a huge role, but it was also a matter of regulatory blind spots. Before Aspen, the primary focus of water treatment was almost entirely bacterial load.
Right, they were just focused on bacteria.
Exactly. The realization that an entire class of pathogens could survive standard chemical disinfection forced the EPA to draft the surface water treatment rule.
And what did that rule change?
It mandates that water systems demonstrate a defined log removal of Giardia cysts.
Meaning logarithmic scales of reduction. So, like a 3-log removal standard means the physical filtration system must successfully trap and remove 99.9% of the cysts from the water supply.
Right. You cannot poison them. You literally have to push the water through membranes that are fine enough to snag them.
So the chlorination handles the bacterial and viral threats, but the physical filtration is exclusively there to catch the indestructible cysts.
Exactly.
And this environmental resilience also explains the famous beaver fever nickname, doesn't it?
It sure does.
Because following the Aspen outbreak, investigators during the 1970s kept finding hikers in the Rocky Mountains contracting giardiasis from remote, untouched streams.
And they trace the cysts back to local beaver populations. Mammals like beavers, dogs, and humans all act as reservoir hosts.
Just perpetually shedding these armored cysts into the watersheds?
Right, where they just wait in the current. It demonstrates a terrifyingly perfect adaptation to the environment.
Absolutely. They deceive us by hiding in plain sight within water that appears utterly safe.
But their talent for deception extends far beyond the environment. For decades, both of these organisms successfully deceived the world's leading scientists who are looking directly at them under microscopes.
Well, the history of their discovery is just a testament to how easily visual evidence can mislead us. I mean, Antony van Leeuwenhoek, the pioneer of microbiology, effectively discovered Giardia way back in 1681.
Yeah, he was suffering from a bout of intestinal illness himself.
Talk about dedication. He decided to examine his own diarrheal stool under his hand-ground lenses, and he noted these tiny moving animalcules.
And modern analysis of his meticulous sketches confirms he was observing Giardia trophozoites, nearly 170 years before Willem Duchamp Lambel officially described the organism in 1859.
Which eventually led to its taxonomic naming after both Lambel and the French zoologist Alfred Mathieu Giard.
Right. And Entamoeba's discovery followed a similar path of intense, albeit ethically questionable dedication.
Oh, Fedor Alexandrovich Lush.
Exactly. In 1875, Lush identified amoeba in a St. Petersburg patient who was suffering from severe dysentery.
And to prove these specific organisms caused the disease, he inoculated dogs with the patient's stool and successfully reproduced the illness.
Which is wild. He was effectively executing the logic of Koch's postulates, the gold standard rules for proving a specific germ causes a specific disease, before those postulates were even fully formalized by the scientific community.
Who wouldn't pass an ethics board today, that's for sure.
Definitely not. But what's fascinating here is that while Lush proved causation, the reliance on visual microscopic evidence eventually laid a massive trap for clinical medicine.
How so?
Well, for most of the 20th century, the diagnostic standard for amoebiasis was morphology. A clinician would take a stool sample, put it under a microscope, and look for an amoeba with this specific shape, size, and internal structures characteristic of Entamoeba histolytica.
Right, the logic being, if it looks like a duck and quacks like a duck, you treat it like a deadly tissue-dissolving duck.
Always the assumption, yeah. But in 1993, parasitologists Louis Diamond and C. Graham Clark totally shattered that paradigm.
What did they do?
They utilized emerging genetic techniques to analyze this single morphological group, and the DNA revealed that these visually identical amoeba were actually two completely distinct species.
Oh, wow. So you had the dangerous invasive E. histolytica and a genetically separate, completely harmless species named E. dispar.
Exactly. E. dispar is a common colonizer of the human gut, but it lacks the genetic machinery to produce the enzymes necessary for tissue invasion. It causes literally zero disease.
But because they look utterly identical under a standard microscope, The medical community had been misdiagnosing and treating them as the same deadly pathogen for nearly a century.
Yes. And the epidemiological fallout from that single genetic discovery is just staggering. Global health statistics prior to 1993 ranked amoebiasis as the second or third leading parasitic cause of death worldwide.
Trailing only malaria, right?
Right. But once PCR and antigen testing, which look for specific genetic sequences or unique proteins rather than just cellular shape, once those were applied to population data, researchers realized E. dispar was vastly more prevalent than E. histolytica.
So they had to radically downgrade the global mortality and morbidity statistics.
Drastically. Millions of patients had been subjected to heavy-duty, toxic drugs to eradicate a harmless lookalike.
That is mind-blowing. Visual morphology created this massive optical illusion that skewed global health policy.
It really did.
And Giardia managed to pull off an equally impressive deception. Right. Not just on clinicians, but on evolutionary biologists trying to map the tree of life.
Ah, yes, the mitosome discovery. It stands as a prime example in biology of a beautifully elegant theory being completely dismantled by a single, inconvenient structural reality.
So for decades, the prevailing textbook narrative, which was anchored by Thomas Cavalier-Smith's Archaeozoa hypothesis, held that Giardia was a living fossil.
Right. Scientists believed it was this ancient eukaryotic cell that had branched off the evolutionary tree long before mitochondria ever evolved.
Because when researchers looked at Giardia's cellular structure, it appeared entirely devoid of mitochondria. So they just assumed it was a pristine relic from the dawn of complex life.
Exactly. It was placed at the very root of phylogenetic trees. It seemed to represent this perfect transitionary phase between simple bacteria and complex cells.
Until 2003.
Yep. That consensus held until 2003, when Jorge Tovar's research team applied more advanced localization techniques.
And what did they find?
They discovered these tiny, highly reduced cellular compartments hidden within the organism, which they named mydosomes.
So they weren't missing organelles at all.
No, they were vestigial, stripped-down remnants of actual mitochondria. The cellular machinery had been there the entire time, just heavily modified and repurposed.
I love the analogy for this. It's like finding a stripped-down car chassis abandoned in the desert.
Oh, that's a good one.
Right. You look at the bare metal frame, completely devoid of an engine block, devoid of any electronics, and you assume it's a historical artifact. You think, oh, this is a carriage built long before the invention of the internal combustion engine.
Because there's no engine to be seen.
Exactly. But then a mechanic looks closely and inspects the frame, and they find microscopic serial numbers for a modern V8 engine. And you realize, wait, this isn't an ancient relic. It's a highly modern sports car that was deliberately stripped of all non-essential weight so it could compete in a demolition derby.
That is exactly what happened. In evolutionary biology, the phenomenon that leads to this kind of misclassification is called long branch attraction.
So Giardia isn't primitive at all.
Not even slightly. It is a highly evolved, deeply specialized organism. It descends from a complex ancestor that possessed full functioning mitochondria.
But it just didn't need them anymore.
Right. As Giardia adapted to its parasitic anaerobic lifestyle inside the oxygen-deprived human gut, it no longer needed mitochondria for cellular respiration. So it ruthlessly discarded that complex machinery to save energy.
It evolved so rapidly and became so reduced that its genetic sequence accidentally tricked our computer models into placing it at the ancient root of the tree of life.
It's the ultimate evolutionary Trojan horse.
But, the fact that these parasites are so adept at masking their true nature creates severe dilemmas for how we actually treat them today, particularly in resource-limited clinical settings.
It really does. The 1993 split between the dangerous E. histolytica and the harmless E. dispar highlighted the absolute necessity of molecular diagnostics like PCR.
But not everyone has PCR.
Exactly. The reality of global medicine is that many clinics in developing nations do not have the infrastructure or funding for advanced genetic or antigen testing. They are still relying on traditional light microscopes.
So the exact same diagnostic trap from the 20th century is still catching clinicians today.
Sadly, yes. Patients colonized with the harmless E. dispar are continually misdiagnosed and prescribed aggressive medications they don't need.
And on the flip side,
conversely, an asymptomatic carrier of the truly dangerous E. histolytica might be misidentified as having the harmless variant, left untreated, and allowed to develop life-threatening liver abscesses months later.
it's an absolute diagnostic nightmare. But here's where it gets really interesting.
Yeah.
The pharmaceutical counterattack is where the biochemistry gets truly brilliant.
Oh, the nitramidazole drugs.
Yes, Both Giardia and Entamoeba are primarily treated with these drugs, the most common being metronidazole.
Hmm.
And the mechanism of action for this drug relies on exploiting the parasite's own survival adaptations.
It's fascinating because metronidazole is a prodrug.
Meaning what, exactly?
In its original administered form, it is completely inert. It circulates through the human host's bloodstream without causing significant cellular damage because it requires a highly specific chemical environment to become lethal.
And that environment is provided by the parasite.
Exactly. Because these parasites stripped down their mitochondria to survive in an oxygen-deprived gut, they rely on anaerobic metabolism. They utilize a very specific electron transport system built around an electron carrier protein called ferredoxin.
Okay, so they have this unique ferredoxin engine running.
Right. And when the parasite absorbs the inert metronidazole, its own ferredoxin pathway inadvertently interacts with the compound.
Oh, wow.
Yeah, the parasite's routine metabolic machinery transfers an electron to the drug, reducing its nitro group. That specific chemical reaction transforms the harmless prodrug into a highly reactive cytotoxic intermediate.
So it basically turns it into a bomb.
Yes, this toxic byproduct rapidly binds to and shreds the parasite's DNA, halting replication and killing the cell.
That is incredible. The parasite's own metabolic engine activates the explosive that destroys it. The drug perfectly hijacks their evolutionary adaptation.
It's brilliant pharmacology.
But wait, if the lethality of the drug relies entirely on the parasite's ferredoxin engine running, can't the parasite just turn that engine off to avoid detonating the drug?
Well, you've just identified the current problem. That exact adaptive mechanism is currently driving a rise in drug resistance, particularly in Giardia strains.
Are you serious? I can just turn it off.
Infectious disease specialists are observing parasites that actively downregulate the transcription of the ferredoxin enzymes required to activate the drug.
So they just stop making the enzyme.
Right. The organisms are essentially starving themselves of metabolic efficiency, accepting a slower growth rate to ensure the prodrug remains inert.
They are literally evolving around our best biochemical traps in real time.
It's an arms race. And even when the drug works perfectly, there is a massive clinical pitfall regarding the treatment of Entamoeba.
Right, because the misconception is that a course of metronidazole constitutes a total cure.
Exactly. Relying solely on metronidazole is a dangerous clinical error. The drug is highly effective at hunting down and destroying the active tissue-invading trophozoates in the mucosal lining and the bloodstream.
So it stops the acute dysentery and the liver damage?
Yes, but it cannot penetrate the thick, chitinous walls of the dormant cysts waiting in the intestinal lumen.
Ah, the hibernation bunkers protect them from the chemical shrapnel.
Exactly. So if a patient only takes metronidazole, the active symptoms will resolve, but the cysts remain perfectly viable.
And then what happens?
Once the drug clears the patient's system, those cysts will eventually exist, hatching into new trophozoites and triggering a massive relapse of the infection.
Oh, wow.
So clinical guidelines strictly mandate a two-step pharmacological approach. The initial metronidazole treatment must be followed by a second drug, a luminal agent like paramomycin.
The 1-2 punch. The first drug neutralizes the active fighters invading the tissue, and the second drug sweeps through the gut lumen specifically to demolish the bunkers.
Precisely. Skip the second step, and you are just resetting the timer for the next infection. Understanding that life cycle is critical for actual eradication, rather than just, temporary symptom management.
Absolutely. Let's synthesize the threat profile we built today. We have Giardia, the flagellate that throws a malabsorptive tarp over the small intestine to block nutrient uptake.
Yep.
And we have Entamoeba, the sarcodine, that actively dissolves the colon and migrates to the liver. They both bypass our multi-billion-dollar municipal water chlorination systems by hiding inside indestructible cyst bunkers.
It's quite the resume.
It really is. Furthermore, they spent decades tricking the scientific community, masquerading as ancient evolutionary relics or hiding behind completely harmless morphological twins.
Yeah, they are masters of disguise.
And for anyone listening, this goes far beyond academic trivia. Giardiasis remains the most commonly diagnosed intestinal parasite in the United States. Whether you are navigating a daycare center or drinking from a seemingly untouched mountain stream, this pathogen is operating in your backyard.
And globally, amoebiasis continues to exert a massive health and economic toll wherever sanitation infrastructure struggles to maintain physical filtration standards.
It's a huge burden.
It really is. Their story is a profound reminder of the sheer adaptability of single-celled life. But, it also highlights a critical vulnerability in how we practice science and medicine, particularly regarding that 1993 taxonomic split of the amoebas.
Right, because we spent a century treating a harmless colonizer as a deadly pathogen simply because our primary diagnostic tool couldn't spot the difference.
Exactly. We placed absolute trust in what we could observe through the lens of a microscope, and it handed us a vastly distorted picture of the global disease burden.
It really makes you think.
It forces you to consider our current medical landscape. I mean, in an age where we are constantly inundated with new medical data, advanced imaging, and novel biomarkers, what other harmless lookalikes might currently be skewing our understanding of human disease, just waiting for the next technological leap to expose the illusion?